All right, hi everybody. Today we're going to talk about Spectradyne's arc particle analyzer and how it can be used in a flow core environment and the value that it brings as an extension of conventional flow cytometry. So thanks very much for joining us. We will have time for questions at the end. So please put your questions in the chat at any time for the meeting, and we'll take them up at the end. Lou Brown, our East Coast sales associate, will handle those, and we can have a good discussion at the end. I look forward to hearing from you about it. All right, so if you haven't heard of Spectradyne before, our mission is to reshape the discovery, development, and production of modern therapeutics. And the way to do that, in the way that we're approaching that, is to develop cutting-edge instrumentation for nanoparticle analysis. So that's what we do. We make nanoparticle analyzers. It's a fun business. The reason that's important is because nanoparticle applications are growing really rapidly. You may work with EVs or extracellular vesicles, LNPs or viruses, or any other kind of nanomedicine, nanoparticles. And these are used or studied in diverse applications: fundamental biology, so we understand what they're doing, investigation of nanoparticles as potential biomarkers, and in nanomedicine as therapeutics. It's really important at this size scale to recognize that the size and concentration of these particles really dictate their behavior in strong ways. So the size of the particles and the concentration determine the dose of a therapeutic, for example. They govern diffusion, so the biodistribution of a drug, or the availability of a nanoparticle in a biological system. And in a therapeutic application, ultimately govern the safety and efficacy of the drug. On top of that, particles are generated now in very complex systems. So we don't make nanoparticles one by one, right? We produce them in complex biological systems often, so tissue culture media, or maybe they're in biological fluids to begin with. And so being able to identify those particles and measure the amounts of active ingredients, their cargo on them, is really important for these applications also. So we've identified, from feedback from users in the market, the size, concentration, and cargo are really three really critical quality attributes for nanoparticles. And I like to illustrate that with a canonical example where in the lab, a scientist is trying to compare two different formulations of nanoparticle therapeutics, or two different samples. And so you've got your samples A and B, and typically the way this experiment is done is you apply these to a test system. Maybe you're just doing downstream analysis, or maybe you're checking their biological activity, like shown here. So maybe that's a tissue culture media or tissue culture model. And A and B affect different changes in that system, and then we analyze that with some kind of downstream analysis. And in this case, I'm illustrating it with a flow cytometry example. So you see a shift in between formulations A and B. B produces more fluorescent cells than formulation A. But nobody here would consider doing this experiment without first controlling for the dose of the sample. It's really critical to know whether you're putting in the same amount of A and B or not, right? If you put in 10 times as much of B, it's reasonable to expect you get a different response in the system. And so it's very critical, really, for basic good science to be able to measure the size, concentration, and payload of these nanoparticles at the input to this experiment. Now, there are a lot of good technologies for measuring the output, right? So flow cytometry and fluorescence microscopy, for example, but there are not a lot of adequate tools for measuring the concentration and the dose of this input material of the nanoparticles. That doesn't mean there aren't technologies out there for doing it, though. So I'm showing you a bunch of them. Dynamic light scattering, really popular in LNP applications. There are surface immobilization-based imaging systems. There's nanoparticle tracking analysis, which is pretty ubiquitous. And there's flow cytometry, obviously of interest to this group. even among flow cytometers now, all the big companies are trying to make nanoscale flow cytometers and to varying degrees of success, but they're essentially trying to scale down the existing technology of light scattering to measure nanoparticles. If you look at all of these examples, what are out there are essentially light scattering instruments. So every one of these technologies is using light scattering to detect or measure the size of the particles. And that technique is fundamentally limited at the nanoscale because basically because of fundamental physics, right? The particles are smaller than the wavelength of light, and so it gets harder and harder to measure them as they get smaller. Then when you do try and use light scattering to infer particle size, for example, you have to make assumptions about the particles that are just not possible to make accurately for heterogeneous samples, and we'll talk more about that in a minute. Because we're a flow cytometry audience here, I'd like to just review flow cytometry and how it's used, try to be applied for nanoparticle analysis. So flow cytometry is a great technique, right? It's been around for a long time. It's a foundational technology for a lot of fields. It's ubiquitous, it's very well characterized. And importantly, we're measuring the particles in flow cytometry in situ, right? They're just flowing in solution. We don't have to immobilize them on a surface first to measure them. And there's a huge amount of well-established protocols and labeling systems for this, right? Just to recap, basically the way it works, right? So particles flow down through a flow cell, and we illuminate them one by one with an excitation laser, or maybe more, right? And then we collect the light, a couple of different kinds of light. So there's forward scatter light, side scatter light, and then there might be fluorescent light emitted from the particles as well. And in a flow cytometer, we detect and count particles using one of two options. One is to detect the pulse in the scatter signals, okay? Or we can detect a pulse in the fluorescent signal, and that's just triggering, the question is what signal to use to trigger the detection event to say, there's a particle, let's analyze it. The difficulty at the nanoscale is when you start counting these particles, okay, and measuring their size. So if we're going to use the scatter signal to detect and count particles, that's an issue because nanoparticles are very small, and a lot of these biological fluid or particles have low index contrast with their surrounding medium. So they don't scatter a lot of light, and that diminishes very strongly with particle diameter. So practically speaking, most flow cytometers limit of detection is about 200 nanometers for a strongly scattering particle, and larger for different weakly scattering particles. The other option, detecting by triggering on fluorescence, really only enables you to detect fluorescent particles for one, so that limits the set of particles that you're detecting in the sample. And they have to be bright enough to be detected, obviously. So what about measuring size? So here we can use either one of those two options. From fluorescence, there are assays where you can calibrate the brightness of fluorescence and assume that it's proportional to the surface area, maybe depending on how it's stained. This is a complex operating procedure, and it still depends on the sample and the stain itself. And the most common way to measure size by flow cytometry is from the scattering signal. So you look at how much light got scattered by the particle, and then you infer the size from that. But in order to do this, and this is the critical point here, in order to do that accurately, you need to know the refractive index of every particle you're counting. And that's not easy. So this plot here is taken from a report, scientific report publication that I'll come back to. This is published by veterans in the flow cytometry field at Beckman-Coulter when they released one of their instruments. And this figure is what I want to focus on. It's showing on the y-axis the scatter in the side scatter channel versus the actual particle size, which was determined with another method for a bunch of different materials and particle types. Okay, so all the green dots are polystyrene beads. Those are ubiquitous, and so you get the scatter versus particle size dependence for polystyrene. And that's very different than these yellow dots, which are viruses, right? And then that's different still from silica, which are these blue particles, okay? And these streamlines that are on here are lines of constant refractive index. according to a certain model, right? And so the main issue here is that if we measure a single particle to have a scattering intensity of, say, 1 × 10⁵ units, and these are arbitrary units, so this is just for a single instrument, we won't know whether that particle is a 100 nanometer polystyrene particle, or a 150 nanometer HSV, or a 200 nanometer silica particle, right? It's impossible to know just from side scatter what kind of particle this is without first knowing the refractive index. And as soon as you start mixing these together or end up in a biological system or even just a complex formulation where the particles have different refractive indices, it's impossible to get an accurate size and concentration distribution from scatter alone, okay? Now, that doesn't stop people from trying, and I just want to illustrate the complexity of this process, because once this paper was published, then shortly after that, another group of flow cytometry experts weighed in and said, no, you guys made a mistake about how you're modeling the refractive index. It's not right. And then these guys, the original authors, had a chance to rebut, right? So this is an ongoing current issue for nanoscale flow cytometry by light scattering. And this is, a lot of complex math, and it's not easy, and all of it breaks apart as soon as you have a heterogeneous sample. Okay, so I don't want to belabor that anymore. The main point is that there's a need for direct and accurate measurements of size, concentration, and cargo, those 3 critical quality attributes for nanoparticles. And that's where Spectradyne comes in. So this is our ARC particle analyzer, shown at right. We launched it about 1.5 years ago. It makes direct and accurate measurements of nanoparticle size, concentration, and cargo starting about 50 nanometers and goes up from there. We're always working on pushing this limit of detection down. Really importantly, there are no assumptions required for the accuracy of this technology. So the accuracy is independent of the refractive index of the particles, their polydispersity, so if you have mixtures of different sizes in the sample, it doesn't matter, or the heterogeneity of different materials in the sample. And I'll show you examples about that today and how it works. On top of that, and specifically relevant for a Flowcore facility, the instrument is fast and easy to use. So it's about 5 minutes of measurement. And importantly, there are no alignment requirements or calibration or cleaning required for this instrument between samples or even regular like alignment or calibration. So all that stuff is automated, and I can explain more about that later if you like. It's also compatible with the flow protocols that you know from conventional flow cytometry, immunostaining and stuff, and I'll also show that. So the way it works is by combining 2 methods. So this is a unique combination of 2 different techniques in a microfluidic chip. So this is an image of our analysis cartridge. It's about a 1/2 inch on the side. And what we do inside here is we flow the sample through a bottleneck in the fluid flow, right? So the particles flow through here one by one in the sample, and every time a single particle goes through, we measure it electrically. So we measure its size and how fast it went through the constriction. So we're not using light scattering to count. We're not using light scattering to measure the size of the particle. This is an electrical method, and it's been around for decades. It's the Coulter counter method, if you're familiar with that. Spectradyne's innovation has been to bring that to practical use at the nanoscale. So this technology, measuring just size and concentration, has been on the market from us in the form of another instrument, the NCS1, for almost 10 years now. What we've done in the ARC has been to couple this with fluorescence of single particles, just like in a flow cytometer. So now we're exciting each particle as it goes through the constriction and measuring the fluorescence that's emitted from the particle in up to 3 optical bands simultaneously with the electrical signal. Okay, so from this single measurement, we're getting an electrical measurement of size and concentration, as well as an optical measurement of fluorescence in up to 3 optical channels. Let me spend a minute telling you about the microfluidics, because they're an important part of why this system works so well. They are highly engineered microfluidic cartridges, right? And this is the main key feature I want to show you. This is an input filter that is embedded into, it's formed in each cartridge to exclude particles that are larger than the sensing constriction. So your first thought was, I've used a Coulter counter at the nanoscale before, and it clogged too much, right? Because there's always big particles in the sample. In our cartridges, we exclude those big particles. We catch them in a filter that's built into every cartridge at the upstream. And so ultimately, what these engineering features mean for you is that you can very quickly and easily analyze very complex samples, and I'll show you examples of that today. And the microfluidics enables the no cleaning, no calibration, and no alignment features of our instrument also, so it enables ease of use. Plus, because we're in microfluidics, we only need a few microliters of your sample to analyze it. All right. You don't have to take my word for this. This technology won the technology showcase at ISAC meeting at CITO last year. Very proud of that. And maybe some of you were there in the flow cytometry community. So thanks. Our key applications for this technology are threefold. We're focusing on nanomedicine, so primarily LNPs, virus and gene therapies. This is basically virus quantification, and also extracellular vesicles. So these are the biological particles that hold so much promise as therapeutics and biomarkers right now. And so I will show you first an example of measuring extracellular vesicles on the ARC. I'll show you 3 examples today, so this will be the first one. Before we get into it, I want to just draw your attention to this recent publication in Nanoletters. This is from Josh Welsh, Joanne Lanigan, and others at NIH. The reason I want to highlight this is because it shows the importance and the power, really, of using orthogonal techniques to measure nanoparticles. So what they did here was combine conventional flow cytometry in the form of the Cytec instrument here with Spectradyne's ARC particle analyzer to derive new information, namely the refractive index of EVs in this sample as a function of size. So this is a cool paper. I encourage you to go check it out. And then let me show you an example of what the ARC can do for cargo analysis using this commercial product. So if you haven't heard of Vesiculab, I encourage you to go and check it out. They have a bunch of reagents and tools for nanoscale flow cytometry and EV research. This one in particular is an engineered extracellular vesicle that is engineered to express a fusion protein between GFP and CD63. So CD63 is kind of a classic tetraspanin protein in the EV world, and they've fused it to GFP to make these particles fluorescent. Okay. When we analyze this sample on the ARC and just look at the size distribution, so this plot is showing concentration on the y-axis versus particle diameter on the x-axis. This is the distribution that we see. Okay, and this is just the electrical measurement data, but we're looking at the fluorescence also. So this is what we see in the fluorescence. So this insert figure is showing a point for every particle that's detected, okay? And we're plotting the brightness in the GFP channel, so one of our fluorescent channels, versus the brightness in our PE channel, this is a yellow one, for every particle that's being detected, right? We have limits of detection like any other instrument, and those are 30 ERF units of FITC in the GFP channel or in the green channel, and just a couple of PE molecules in MESF units in the PE channel. Okay, so we can be quantitative about the brightness of these particles, and as expected, right, these are unstained samples, but they express the GFP. We see a bunch of fluorescent green ones here. It gets more interesting when we come in with an antibody now for CD63. So the staining is very easy. You just mix with the stain, and there are no washing steps to reduce autofluorescent or background fluorescence like you would maybe in a flow cytometer. What we see is that the particle size distribution does not change significantly, which is what we expect from tacking on a few antibodies onto these particles. But the fluorescence changes significantly. So what we see here is that pretty much every time there's a GFP on the particle, it also has CD63, right? So there are no particles essentially that have only GFP without the CD63, picking up the CD63, which is what we expect because they're fused together, right? There's also a population in here that pick up the CD63 antibody because they're expressing it, but are not GFP fluorescent. And these are the wild type CD63s that are still expressed on some of these particles, right? Because it was not a complete knock-in of the GFP fusion protein gene. So this is cool, and we can analyze any quadrant analysis. We can perform different gates, custom gating here, and always for any gate we can plot the concentration versus real particle diameter here, and that's what's shown over here for the different quadrants in the scatter plot. If we focus in on the double positive population, we can go a step further and ask, well, how bright are these particles? Okay, because our fluorescence measurements are quantitative. And we know, so this is the distribution of brightnesses, so concentration versus brightness for that double positive population. And we know what these units are. They're real units. They're not arbitrary units. Our system is calibrated to report MESF units. So what this means is that there's a population here that have, on average, about 3 PE molecules per particle on them. And because we know it's about 1 PE per antibody, we can say that there are about 3 CD63 epitopes per EV in this sample. So this gives you an example of the kind of multi-dimensional analysis we can do of nanoparticles like EVs with different colors, different channels, right? It's very direct and it's accurate sizing. It's quantitative in every dimension that we report data in, right, in real units. We've quantified surface marker expression, and it's fast and easy to use. So every one of these measurements, really there's only 2 in here, took about 5 minutes to complete, okay? So let me shift gears now and show you my second example, and then we'll do one more after this. This is taken from the virus application world, and as we know, there's a lot of overlap between nanoscale flow cytometry, EVs, and viruses, so I thought this could be interesting. Just as context, we have viruses are great samples for us because they're beautifully well-defined particles, right? And so over the years, we've collected many examples of viruses being quantified using our instruments, and these are just some of them. So if you don't see your virus on here, feel free to reach out, and we'll love to talk about that. The example I'm going to show you today is immunostaining of a lentivirus sample to look at the number of expressed copies of this fusion protein that is engineered to express on the surface. So in order to prepare the sample, it's very easy. We put in the primary antibody for the surface protein that we're looking for, and then come in with a secondary antibody conjugated with Alexa 488. And we just mix and measure the sample. There's no sample cleanup required to reduce autofluorescence or fluorescence in the background, excuse me. And we also ran a control sample where we omitted the primary antibody so that we can show that the labeling is specific, that we measure these on the arc. So again, here is now another scatter plot. So we've got the brightness on the y-axis and the true particle diameter on the x-axis. And you can see that there's a cluster of particles here. These are the virus particles at just the right diameter for this lentivirus, right? And we can be quantitative over the different quadrants. So above our detection threshold, the concentration of virus that are fluorescent are 1.3 times 10 to the 11 particles per mil. And that represents 43% of all the particles in the sample. There are a lot of other materials in here. So that's one example of how we can measure the size and brightness of these. But because the fluorescence is quantitative, right, we can look at the distribution of brightnesses, and we can see the virus out here as a clear standout population in the sample. And so we can measure that there are about 200, the equivalent brightness of 200 FTSE molecules per particle in this population. And so that allows you to, in a virus production environment, to compare different formulations and understand how well your virus is expressing the fusion protein on its surface. Okay. So here's an example that shows how the ARC was able to quantify this pseudotype lenti for concentration, size, and the abundance of surface-bound fusion protein in a very fast, easy workflow. Right. As a final example, I wanted to show you how the ARC can be used to quantify particles, nanoparticles, in extremely complex samples. And so what we're going to do here is simulate as an environment where you've got plasma and a fluorescent subpopulation in there, and ask how well can we quantify that fluorescent subpopulation in the mixture. So what we did was we took plasma and we spiked in at a serial dilution of concentrations, 100 nanometer fluorescent particles, these are just polystyrene ones, into the plasma. Okay, then we measure each sample on the ARC. What I'm showing you here now is the particle size or concentration on the y-axis versus size on the x-axis for the mixture for two different populations. In blue is showing the distribution of all the particles in the sample, fluorescent or not. And in green is the particle size distribution of just the fluorescent ones. Okay. And so what you can see is at this very high spike in concentration, the fluorescent particles are prominent enough in the overall size distribution that you can see they form a bump in the overall particle size distribution, right, at 100 nanometers. But as we dilute the sample out, or as we dilute the fluorescent particles down, you can see that they become less and less of a feature in the overall size distribution until they can no longer be visible. They're a minority population in the mixture, right? And so this is just, we're running, this is a measurement of 6 different samples, plasma mixed with these fluorescent particles at decreasing amounts of fluorescent concentration. And you can see that we're still able clearly to detect the fluorescent particles in this mixture down at much lower concentration than the surrounding plasma. There are a bunch of different ways to look at this data, right? So in this first plot, this is just a total particle size distribution for each of the serial dilutions. And you can see they stop changing once you dilute far enough, right? In the fluorescent scatter plot now, you can see how the fluorescent particles stand out from the other particles. They're above the fluorescent detection threshold for this in this measurement, right? And all the plasma particles are down here, non-fluorescent. This is the size distribution of just the fluorescent ones. And then we can plot the concentration as a function of dilution factor. And you see that we get a nice linear response all the way down to almost 100-fold lower concentration than for the plasma alone, right? And so this shows that we can accurately quantify fluorescent subpopulations in samples as complex as plasma directly in that biofluid. And there are opportunities to avoid use of animal models this way because it enables more in vitro testing actually in PK studies of nanomedicines. All right, so those are the data examples I had prepared. And so in summary, the ARC measures very directly and accurately nanoparticle size, concentration, and fluorescent cargo from about 50 nanometers and up. No assumptions required for the accuracy of the measurement, and it's very fast and easy to use. It's a good fit for a flow core because of a number of reasons. It's a natural extension of flow cytometry to the nanoscale, right? Where it's compatible with all the flow protocols that you're used to using on a flow cytometer, but be able to extend the sizing and the detection down far below the limits of conventional flow cytometry. The ARC offers new capabilities for a broad user base. So we showed you examples of EVs and virus. We've got lots of data for LNPs, many materials. available to be characterized on this instrument. It's easy to adopt because it's compatible with what the staining protocols you're used to working with, and it's very practical and easy to use. I would encourage you, if you'd like to learn more, to first of all ask questions here, but also visit our website. We have a bunch of information there. You can go directly there if you scan this QR code right now. We have a lot of other information available on our website, like recorded webinars. We'll be posting this one again later on our website, but also past ones. Actually, this is especially relevant for the flow cytometry community. This webinar included a presentation from the Mayo Clinic where they compared the fluorescence or the sensitivity and the performance of the ARC to conventional flow cytometry and presented a bunch of data in this webinar also. So please come visit us at the website, and thank you very much for your attention.